Hello, everyone. Thank you for joining this installment of Boyce Thompson Institute's new Breaking Ground discussion series. My name is AJ Bushy, and I am in the Communications Department here at BTI. And for this month's Breaking Ground, we welcome Dr. Maria Harrison, a plant molecular biologist here at BTI, who specializes in the symbiotic relationships of plant roots and soil fungi. And today we'll be talking about some of these symbiotic relationships and how they could be used to improve agricultural practices, particularly by helping reduce the amount of fertilizer used. Hi, Maria.
Hey, AJ.
And before we get started, we got a couple of housekeeping announcements. During the session, all participants will be muted. And we do, this is like an hour. We'll hopefully be done by 1. And we do about a half-hour discussion with Maria and myself, and then about a half-hour Q&A. So I'm sure everybody here knows how Zoom works by now. We've all been doing it a while. You can ask questions in the chat, and there's also like a little Q&A button down below. You can put your questions in either spot, we'll get them. And then we'll do the Q&A session for about the last 20 or 30 minutes or so. And thank you all again for joining us today. Appreciate it. We got a good crowd today. And so I guess to start things off, Maria, could you tell us a little bit about yourself and your research and kind of how you got into what you're currently doing?
Certainly. I will just share my screen because I have made some,
Sure.
Let's see. Aha. Can you see that?
Yes, I can.
Great. Okay. So just get rid of some of this stuff at the top. Yeah, so AJ asked how I started, and so these many images are giving you a path through my training and what led to the work that we do today. And it started actually over here on the left. I was an undergraduate studying microbiology at the University of Newcastle upon Tyne, and I was fascinated by microorganisms and what they did to, particularly at that time, to animals and humans. And this was also the time that I got my first research experience in a lab. I worked as an undergraduate I did a project, an honors project, and I also had an internship in a company that made restriction enzymes. And those things made me interested in molecular biology. And so then for my PhD, I moved to Manchester and worked with tomatoes actually and studied seed development, but particularly using molecular biology approaches to look at genes that were expressed during seed development. And so that got me interested also in plants. And then for my postdoc, I decided to combine the two, and I joined Rick Dixon at the Nobel Foundation to study the interaction of plants and pathogens. And it was actually working with beans and with a fungal pathogen, Colototricum, but we actually didn't touch the bean or the Colototricum fungus, because there was a much more precise way of doing it, which was to have the beans growing as cells, single cells in culture, and to take the molecules from the wall of the fungus and apply them to the beans, and then study what the bean cell did in response. And basically the bean cell turned on a lot of genes that made secondary metabolites that defended it against the pathogen. So this was my first interaction sorts of studies. And having done that, I was fortunate to be offered a group leader position at the Nobel Foundation, and I wanted to continue on plants and microorganisms. And so we come over here, and I started the studies of plants associating with fungi, this time not a pathogenic interaction, but a mutualistic interaction that has benefit for both the fungus and for the plant. And so you'll hear more about this shortly. I also wanted to just say here that I've worked on this ever since then with many, many different people. And the images on the right show some of the people who've been in the lab over the last few years. And I think that's a really nice point about science that maybe I didn't appreciate when I started as an undergraduate, that if you stay in a research career, it's really an international endeavor, science. And so you have the opportunity to work with people from all over the world and also at many stages of their careers. And so that's also very fun and exciting. And you see some of the people here in this slide.
Yeah, that's a great point. And you know, in today's day and age, there's so much more of that. I mean, we had so many registrants from all over the world, India, China, Mexico, all over the place. So it's pretty good stuff. Just one quick question before we move on. Was there anything in particular that led you to switch from like disease causing microbes to like this more symbiotic relationship?
Actually, yes, which I can show you a little better maybe on the next slide. I can tell you what really started me on that. And it was really images like these. So I was listening to a talk from Professor Larry Peterson, who is a professor at Guelph in Canada, and he was giving a talk on mycorrhizal associations. And this is what you're seeing on the screen here now. And I'll tell you more about some of these images in a moment. He was showing very similar images, and he's a cell biologist and had studied these associations for many years. And he made the point that molecular biology was coming of age, and it would be a good time to try to apply some of those techniques to mycorrhizal associations and learn the molecular basis of these plant fungal associations. And that's what really, it was the images and his talk that really started to catalyze my interest in those interactions. And since they were very understudied and there was an opportunity to do that, I thought, well, this is where I can make a contribution. I can apply my molecular skills and see what we can learn about this plant fungal mutualistic association. So that's really how I switched.
Yeah, you always have the coolest pictures. I always love working on your stuff because they're always gorgeous pictures.
I can tell you a little about what this is. So if we start over on the left, we're working on something that occurs underground. So this is an interaction between a fungus and a plant, actually very widespread in the plant kingdom. More than 70% of the flowering plants will have the capacity to form associations with mycorrhizal fungi, arbuscular mycorrhizal fungi. And the little yellow spheres that you see here are the spores of the fungus. But if you look at this picture otherwise, you may think that there's really not much going on. You can see some spheres and you can see roots. But that's reality. That's really how it is. If you want to really know what's going on, you have to look deeper into the roots with microscopes and stains and also molecular techniques. And so if you move across to the next image, this is the roots now after you've washed them carefully. And what you can still see is not too much, a root, and also these strands that go out into they would be out in the soil, they were hidden on here. And these are the hyphae of the fungus. These are filaments of the fungus that grow out of the root into the soil. You still don't see too much. And if you stain this root and use a microscope, you can move to this image and you can see that this is an image of a root, and these are all root cells. And what you can see here, all this pink material in the middle, is the fungus. So it's the fungus is living inside the root and actually inside the root cells. And these pieces down here, these strands, are the ones that you could see out here. So those are going out into the soil. And still, you can't really see what's going on in here. Looks a bit fuzzy. So if you increase your magnification again and change microscope, go to a confocal microscope or a scanning EM microscope up here, then you can see really what the fungus looks like inside the plant cell. So you can see all these sort of branches, fungal branches. These are also hyphae. They're very branched hyphae, and they have a name, which is arbuscule. It comes from the Latin for small tree. And some people think they look like trees. I think, yeah, some of them look like trees. This one looks to me like a coral, I think.
Yeah, I think they look like trees. I can see that.
Yeah. Yeah, so this is the fungus inside the plant cell, the root cell, and all of these cells that you would see here in pink have one of these inside them.
That's very amazing. So what exactly are they doing in there?
Yeah, so the purpose for this association, the reason for each organism to interact with the other, is that they have the capacity to provide each other with something that each of them needs, something different. So the fungus needs carbon, doesn't have a way to break down organic matter in the soil. It needs to be fed carbon, and it particularly needs to be fed lipids because it has no way to synthesize its own lipid. So its purpose for colonizing the root is in order to get carbon, and particularly to get carbon in the form of lipid. And it gets that at this interface with the plant cell. The plant cell provides lipids and the fungus is able to take them up. But the fungus also provides something to the plant, and this is important too. And the fungus is providing the plant with mineral nutrients, in particular phosphorus, which it provides as phosphate. And the way it does this is these external hyphae that are external to the root out in the soil, they have the capacity to take up phosphate from the soil, and it moves through these strands. These strands are all connected back to the internal structures, and the phosphate moves through the system to these internal structures, the arbuscules, and then it's released to the plant cell. And so that interface is very important because it's the site of the exchange of nutrients between the two organisms. Phosphates coming out and going into the plant cell, and carbon in the form of lipid, and probably also sugar is going into the fungus. So they make this association in order to get mineral nutrients from each other, to trade mineral nutrients. And they do this, particularly the plant does this when it's in a low phosphate condition, when it doesn't have enough phosphate.
So it's kind of interesting that the fungi can't make fatty acids, right? Isn't that strange? I mean, have they always been that way and needed to get the fats, to get the lipids from somewhere else, or did they like lose that pathway somewhere along the way?
Yeah, so we don't really know when they lost it, but all of the fungi, all of the mycorrhizal fungi, or arbuscular mycorrhizal fungi, these are from a subphylum, the Glomeromycotina. All of these fungi that have been sequenced to date or examined to date have lost a very highly conserved enzyme called FAS, fatty acid synthase. And because of this, they're incapable of making their own fatty acids. So we haven't sequenced very many. The field, the research field, haven't sequenced very many arbuscular mycorrhizal fungi, but it's predicted that they will all be what we would say fatty acid auxotrophs, incapable of making their own fatty acids.
Well, that's interesting. And so plants do have immune systems, right? So a lot of fungi and bacteria and viruses will turn on that immune system. Is there something special about these for, from a molecular biology basis for why they don't turn on that immune system?
Yeah, so this is actually still an open question, I think only partially answered in the field, how these fungi manage to either avoid inducing a substantial immune response in the plant, or have a means to either suppress it or to induce the plant to suppress it. We know that when the fungus first starts to colonize the root, there is a small immune response in the plant, but then it's very rapidly dampened. But we don't know the molecular basis of really how this occurs. And it seems at the moment that some of the immune signaling is probably important for starting the association, but exactly how that becomes how that enables a mutualistic interaction and not a pathogenic one is still unknown.
Okay. And so you had some other cool pictures, if I recall. And
I can just flip over to the next one. This was just to really address a question, which is why is the plant not capable of getting enough phosphate by itself? Its roots can take up phosphate from the soil, we know that. So why is having a fungal symbiont particularly beneficial? And the answer is really because of the problems of getting phosphate. So phosphorus is a very reactive mineral and reacts in the soil with other things to make complex forms that don't diffuse through the soil. And the form that the plant can take up, and also that the fungus can take up, are the ionic forms of phosphate, free ions. And these are very scarcely available in the soil. And they also bind to clay, so they don't really move through the soil very well. And what happens around the root is that the root takes up phosphate in its immediate location, and this leaves a zone around the root that's depleted of those phosphate ions. And it takes a long time for more phosphate ions to diffuse in. And so what the root does is grow through the soil to try and find new areas to mine for mineral nutrients like phosphate. But the other way to do it is to take advantage of your fungal symbiont, and those hyphae spread out beyond the depletion zone and capture mineral nutrients like phosphate that otherwise the root wouldn't be able to capture. And they're particularly good at it because they're much finer than the root. So they're able to get into small pockets in the soil that are not accessible to the root. And you can see that if you look here really how how much difference there is in the size of a sorghum root and to the size of a hypha. They're really, really tiny, so they can really invade and have a lot of capacity to capture things from the soil. And if you look at the one at the bottom, it's to kind of give you an idea of the scale. The brown thing on the top is a human hair, and the green thing on the bottom is a hypha that's been stained with a dye to make it green, and you can see the difference in size. So it gives you kind of feel for how fine these hyphae are, and therefore they could get into tiny spaces in the soil.
So they even get into clay?
They can get through clay.
Wow.
They can get through all sorts of particles into little cracks that the root wouldn't ever be able to get into. And in those little cracks and crevices, they're able to scavenge any free phosphate ions and then take some of those back to the plant.
So is there some sort of like signaling pathway that they can signal to each other in the soil?
Yes, so that's comes to this question of how do they really start to interact? How does the fungus find the plant, and how does the plant find the fungus? And it's through chemical signals. When the plant is starving for phosphate, it secretes complex molecules called strigalactones. These are plant hormones, and this was shown some years ago by Koki Akiyama's group in Japan. And these are signals, these can be perceived by the fungus. And once the fungus sees this molecule, it activates its metabolism, starts to grow more rapidly, and it's a signal to the fungus that there's a root very close to it. And so once the fungus starts to ramp up metabolism and grow close to the root, it too secretes molecules that are signals that the plant can perceive. And these are chitin molecules with a lipid chain. They're called lipocyto-oligosaccharides. And the plant perceives these. And when the plant perceives these, then this triggers changes in gene expression in the cells underlying where the fungus is close to the root. And these changes in gene expression change the interior of the cell, and the cell starts to build compartments that allow the fungus to get into the root. And I can show you a very nice animated model that was made by a colleague in Italy, Andrea Genre, and he published this some years ago now in Plant Cell in 2005. And it's a model showing you the sorts of changes that occur in the cell when the fungus is coming onto the surface and then growing into the root. And so I'll play this for you, and you'll be able to see sort of what happens. So the fungus is coming in, it's pink, the pink thing here, and it's signaling to the plant with its lipocyto-oligosaccharides. And this causes changes in this cell underneath. And some of the changes are that the organelles move around, so that's the nucleus moving towards the fungus. And the other change that's triggered is that the cell starts to build a membrane-bound tunnel through the cell. And that's what you can see happening down the middle there. The membrane is in yellow. And this tunnel then spans the cell, and the fungus grows through this tunnel, so it can cross the cell. You can see the fungus now growing into the tunnel. And since this is a cartoon of the cell that's on the outside of the root, the fungus is going to go straight through, come out the other side. But if you can imagine the same thing happening inside, in those cells inside the root where there was the very branched hypha, well, not exactly the same, but a similar thing happens in those cells, and the plant builds a membrane that surrounds all of that branched hypha inside the cell. It's a pretty nice model, I think. Yeah,
so they're not quite exactly inside the cell. They're just really, really close.
That's correct. So yes, if we just pop back to this one, this image down here, you're actually seeing, you think you see a green stained fungus, but actually what you're looking at is a plant protein that's in that membrane around the fungus. And the membrane so closely fits around the fungus that you really see the shape of the fungus. It's like having your hand inside a glove. It's completely surrounding it. And so the green here is not actually the fungus, it's the plant membrane. And
you're seeing the glove.
You're seeing the glove, and the fungus is inside.
Yeah.
This is actually a live image, which is something you can do with the confocal microscope.
Very neat. Very neat. So how old is this relationship? You mentioned, over 70% of flowering plants have it. How old is this evolutionarily?
So very old. There's good molecular data from sequencing and also fossil images that would say that plants have associated with arbuscular mycorrhizal fungi for over 400 million years. So really when plants first started to grow on land to colonize a terrestrial environment. And the images over here on the left were given to me by the late Professor Tom Taylor from Kansas University. And these are polished rocks that show the fossils of ancient arbuscular mycorrhizal fungi inside a now extinct plant that was called Aglaophyton. And so you can see here some hyphal strands and things that were very likely ancestral arbuscules inside the root, those branched hyphae. So it's thought because of this, because of their existence very early and because of their beneficial effect on the plant and their ability to help the plant get mineral nutrients from the soil, it's thought that they may have been very important in this first colonization of terrestrial environments. And particularly because the early plants didn't really have root systems. They had something called rhizoids. You can see here, this is the Aglaophyton plant, and this is what would be its kind of primitive root system. It's not really roots, they're rhizoids, but you can see that they're very small. And if you have to capture things from a tough environment with such a small root system, sort of makes you realize that maybe having a fungal symbiont would have been very helpful. So probably they were instrumental in the colonization of land, although this is still obviously a prediction because we don't know for sure.
Yeah, but some of those early some bryophytes today still have them.
That's right. So some of the most primitive or early diverging land plants, the bryophytes, still have the capacity to form associations with these fungi. Should say there are some other fungi that were around at the same time, and it's likely that they were important too. But certainly mycorrhizal fungi were part of this, I think.
So this thing looks giant in the middle. Is it really?
Yeah, it's funny because this is
big as mountains.
This is an artist's rendition. It's nice. I like it. But actually, if you look up what the predicted size of these Aglaophyton plants were, they're basically not more than 6 millimeters in diameter, these stems. So 6 millimeters is very small and probably not bigger than your hand high. So not quite as dramatic as the one shown here in the image. But, you know, some artistic license is allowed.
That's okay. That's okay.
Yeah.
So why again, and I guess in the question, why don't all plants have them? Has this been lost somewhere along the way, or like different fungi have kind of taken over the role, or?
Yes, combination of both. So these were the earliest associations, but then other mycorrhizal associations developed as well. As one ectomycorrhizal associations with different sets of fungi, orchids. And so in some cases, different plant groups lost this particular association, but gained another one or developed another one. And some lost associations completely altogether. And like the Brassica family, they don't form any mycorrhizal associations, but they do have root endophytes. And some of those endophytes can also contribute the similar sorts of things, like assisting with the capture of mineral nutrients.
Interesting. And so you mentioned the Brassicae, which is a huge family of crops. We know plenty of crops in that family. And so how could the symbiotic relationship be used to help agriculture and the environment?
Yeah, I can give you another image here. So if the plant's growing in a low phosphate environment, then usually these fungi are beneficial. And you can see this here these are in the BTI greenhouse, or they were in the BTI greenhouse. They're sorghum plants. And the ones on the left are growing in pots with no fungus. And the various plants to the right are growing with different fungi or different combinations of fungi. And you can see, I think in general, that growth is promoted when there's a fungal symbiont in the soil compared to the controls without any fungus. But you can also see that there are differences in the amount of growth that's promoted depending on which fungus is in the soil or which combination of fungi. This one called GV promoted growth very well, but then when it was with another one, and there's another one here, GC, was promoting growth well. But the two of them together really didn't have a very beneficial effect on the plant. So we know that there's the capacity to stimulate growth, but the outcome of the association, whether or not it's really beneficial, depends on the fungus. It also depends on the environment, and it depends on the genotype of the plant, which is not shown here. These are all one genotype. But if I would show you a number of sorghum genotypes, they would respond differently to different fungi. And so we don't know the basis of that. But I think if we can understand this type of molecular basis, it should be possible to breed for crops that are optimized to be able to get mineral nutrients via the symbiosis. And this could help capture phosphate from the soil, would improve fertilizer capture. And the reason this is particularly important, capture is important, is that at the moment only about 10% of the phosphate fertilizer that's applied in a given year is actually captured by the crop. And some of the rest of it, either it's bound in the soil into forms that become somewhat insoluble, maybe they're captured later in subsequent years, but some of it runs off into aquatic waterways and leads to pollution problems. And these are images of one that happens in the US every year in the Gulf of Mexico. You get this runoff, which comes from a lot of it from agricultural land running off into the Mississippi watershed. Some of it's also from cities. And this eventually discharges into the Gulf. And this large amount of phosphorus and nitrogen that's in the water causes algal blooms. And these cause oxygen deficiencies in the end. The algae bloom, they sink to the bottom, there's an oxygen deficiency, and it kills off aquatic life. And so if you could capture more efficiently the fertilizer that's applied, and possibly even apply less, this would have beneficial effects for the environment.
Yeah. So real quick, you said only about 10%?
That's what the estimates are. There are published estimates that say only about 10% of what's applied is actually captured. Wow. I think it's going to depend on a lot of different things. It's going to depend on how it's applied. It's going to depend on whether it would rain hard after the application. A lot of different factors, but one general study suggested only 10%.
Yeah, I would assume there's other things in the soil too, right? There's bacteria and all kinds of other
things are going to capture it as well. Yes, it's not just for the plant.
Right. So I can imagine this getting pretty complicated to figure out what exactly is the optimal condition for this particular genotype of this particular species.
Yeah.
And to spread that around for every genotype of every crop could be.
Yeah, it could be complicated.
Yeah. That would be an undertaking. And I would imagine also different methods of doing agriculture, like a tilling. I've a really interesting lecture at AAAS last year at the annual meeting, and I forget the researcher's name, but she was talking about how tilling can interrupt these networks of the fungal networks and that could, and the bacteria as well, and that could also impact how much fertilizer gets taken up, how much of these nutrients get taken up.
Yes, and definitely impacts the health of the fungus. If you break too many of the networks, then you not only break that uptake capacity, but if you sever them from the root is their lifeline for carbon. So they need that carbon to be able to sporulate and form the next generation of fungus in the soil. So it's also important for keeping a healthy population in the soil, and not if you would repeatedly till, you would start to reduce the populations in the soil.
Yeah, very, very, very complex. Okay, I think we have time. We were going to talk a little bit more. We've seen a lot of really cool pictures.
Actually, I was just going to show you one that I didn't show, which was coming from this. I should have moved on from this, but another reason why you don't want to lose too much phosphate in any form is that actually it's a finite resource. So the phosphate fertilizer that's used comes from rock, phosphate containing rock, which is mined usually with strip mining from the earth. And so it's like oil. It's a finite resource, and at some point it will be depleted. So it's important to use what we have efficiently in order to be able to have phosphorus fertilizers in the future. The amount of the reserves is difficult to estimate. So the time frame for which they will be consumed varies depending on whose estimates you read. But I think one certainty is that it is a finite resource and it has to be conserved and used efficiently. And this is kind of an amazing picture. I think it's phosphorus rock mine tailings. So all of these looks like little sand dunes is the remnants after the phosphorus has been mined out. And to give it some scale, if you look down here, what looks like a small child's toy is actually one of those enormous excavator type of cranes. And so I think it illustrates the extent of the mining very well. And this was an article in Nature now in 2009 that started to worry about or discuss the problem about phosphorus reserves and conservation of these resources.
Yeah, that's a pretty crazy picture.
It is.
So you use a lot of cool microscopes in your work. Could we talk a little bit about them and see some more?
Yes, we do. We use microscopes a lot. So we have some different ones here. This is a little lab microscope that we use just for sorting the spores of the fungus in order to get the spores so we can set up our experiments. This gives you a low power magnification, but it's good enough to sort the spores. And then the one over here is another what we call stereoscope, and it has fluorescence capacity. So you can shine UV light onto samples and they'll fluoresce, and we can see that fluorescence. And that's useful for us because some of the stains we use are fluorescent, and so we can see the fungus with these different colored stains. This one is a stain called Alexaflor 488 that gives us this green color. And you can see that the magnification's enough that you could see the fungus inside the cells and some of those hyphae, but you're not able to see distinct features. And if you come down to the confocal microscope, which is here at the bottom, this is also able to image fluorescence, but the resolution is much higher. And so if you come over to a picture here, those blocks that you see up here are actually resolved much better with a confocal microscope. You can see here, you see all the little branches of the fungal hyphae. So a much higher power magnification going on. Plus this microscope can image multiple colors at the same time. So here we've been able to stain the roots with a red stain to see the walls of the cells, of the plant cells, the root cells, and the fungus is in green. In this microscope, we can't do a double image. So you can only see one color at a time.
And these are all here at BTI, right?
These are all here at BTI. And this is actually, sadly, our poor confocal microscope at the moment. In the era of COVID, we have to cover everything with cling wrap between users, so nobody can touch the microscope with their eyes. So you have cling wrap on the eyepieces and cling wrap, well, we took it off right now, but there's cling wrap on the knobs and everywhere else as well. But at least we can still image even if we have a pandemic. And one nice thing that this machine can do, it can image live samples. And so that allows you to see, you can label cells with fluorescent proteins, and you can see things going on inside the cell in real time. Well, actually, I don't think this is in real time, but you can see things going on in the cell and movement within the cell. And this is an image of a component in the cell called the endoplasmic reticulum. It's part of the machinery where lipids are synthesized and also membranes are made, and it moves. And you can see here in this image that the ER is how the ER moves in a living root cell. And you see it moving a lot of stuff around in the cell. We don't know.
So is that the kind of dark red thing in the middle, is that the nucleus, or is that?
I think it is. Let's have a look. Yes, it is, because the ER surrounds the nucleus. There's a part of the ER called the perinuclear ER, and that's what the ER is showing you there in the middle. It's going around the nucleus.
So they're making the lipids.
I mean, they're probably making lipids in there, yes. Yeah.
Okay.
And other things.
Very cool. All right. Looks like we got a lot of questions. Let's move on to some questions.
So I'll stop the sharing. Have I come back to? Yeah.
So from Josh Judkins, is there any exchange of genetic material, RNA or otherwise, between the intracellular fungal hyphae and the plant cells?
That's a good question. I think at the moment, we don't know if there's continual exchange of nucleic acid, but there's certainly evidence that the organisms have swapped genes in the past. But whether there's active transfer of small molecules between them it's not so clear at the moment. There's some evidence to suggest it.
And is the intracellular plant membrane hyphae interface similar to a synapse? It's just kind of a similar question. Sure.
It's certainly it's similar in that there's a lot of membrane and certainly signals are transmitted between them, but the structure is quite different. Similar to a synapse, they have a lot of channels and proteins in the membranes that are moving ions between the organisms, or that both organisms can access. And so in some ways it's analogous, but it's not doing the same sort of rapid transmission of signals that a synapse is doing.
Interesting.
I'm just trying to get out of this PowerPoint presentation, sorry. That's because it was blocking my view. Okay.
From Stephen Rossthorn, does the internal plant structure contain greater amounts of phosphate uptake transporters to enhance the uptake from the arbuscule?
Yes. So that actually the green, the picture that I showed you where we were looking at the membrane around the fungus, the green fluorescent protein there is tagging a phosphate transporter that's in the membrane around the fungus. So you were looking at the phosphate transporter protein in that image. And
so that was the glove, when we're talking about the glove?
Yeah, the hand in the glove picture, yeah. The glove was actually a GFP labeled phosphate transporter.
From L. Longchamps, are mycorrhizal fungi species very specific, or can they grow in symbiosis with different plant species?
Yes, they tend to be very non-specific in their ability to interact with different plants. So many different plants can form associations with many different fungi. But as I showed you in that sorghum picture, the functional outcome for either the plant or the fungus is different in different cases. And so it may be that it really, if you were out in an ecosystem, not just putting things together in the lab, that you would see preferences. But if you take a lot of fungi and you put them with a lot of plants in the greenhouse, you're probably going to see colonization of all the root systems by all the fungi to some extent. But there'll be differences in the functionality.
Very complicated.
That answer the question.
Okay, here's a good question. Stephen Rossthorn, if soil management is critical to maintaining a healthy population of microorganisms, soil be, quote, seeded to enhance particular groups, including the arbuscular mycorrhiza?
Yes, I think that's what a lot of people are interested in, and certainly what a lot of companies are interested in as well. Can they make inoculants that are useful? And it's a complicated thing because there's a lot of different aspects to whether or not they're going to be beneficial in the particular soil with a particular plant species, and also whether they're going to persist, whether they can compete with whatever's in the soil naturally. So it's a very complicated thing to address. And obviously a lot of companies are hoping that you can make inoculants that will be useful. But there's also been a lot of examples where things inoculated in are very soon competed out. And so I think probably the inoculants have to be a complex mixture themselves, and they have to be robust against other organisms.
There's another related, similar question: is inoculation with AM fungi or signal molecules more effective as a treatment for enhancing phosphate efficiency? So I guess you could use signaling molecules instead of.
Yeah, perhaps if you had exactly the right ones and were able to, I think it's probably extremely expensive to make them in the amounts that you would need for agriculture. You could, the other difficulty with the AM fungi, and something I didn't mention, is that they're obligate biotrophs. This means that they are, well, I said they were dependent on the plant for lipids, but there's no way to grow them without the plant. So nobody's been able to grow them by feeding them lipid on a plate or in a culture flask, not like those beans, nice bean cells that I showed you earlier. Which means that if you think about making inoculum, it's quite challenging. You need to have your inoculum produced on a live plant or live plant roots. So it's not so trivial to do.
That's interesting. Do AM fungi, are they capable of impacting root architecture?
Yes, they do change the architecture of the roots. There are several publications that report this. They seem to promote or change architecture such that the root system will make more lateral roots, the small side roots. Definitely have an impact, and that changes the plant's capacity to absorb things from the soil as well. It changes how the plant grows.
So when there's more of the AM fungi, they're more likely to go lateral.
They at least in the, that there's probably differences in how different plant root systems respond, but in the ones that have been examined in the lab, then they're causing more lateral root growth.
Interesting. Do plants like spinach that are not colonized by AM fungi, is there an advantage or disadvantage to the fungi or plants, or is it a defensive strategy? Can AM colonization be pathogenic to any species if it induces an imbalance between carbon, nitrogen, or phosphorus?
I think there's certainly examples where colonization doesn't lead to a growth promotion, maybe to a negative growth response, a growth suppression. There are certainly examples of conditions where that happens. I think that's not the case for the spinach, because spinach actually isn't forming any association at all. And it's not clear why the brassica lost, the brassica family in general, why they lost the ability to form mycorrhizal associations, but there must have been at some stage a negative effect of having the association. So it was advantageous to ditch it. And then that happened. And they must have taken other strategies to be efficient in capturing mineral nutrients. And it's not clear what that is. They have very fine root systems. Some people will suggest that that's their strategy, or that they are annuals that flower very quickly, and their response to low phosphate is to flower and bolt and go to the next generation. But there are other species that form mycorrhizal associations that have the same fine root structure. So I'm not sure how the brassica cope with not having AM fungi. But they do have, as I said, they do have other root endophytes. So AM fungi may be not important, and maybe another root endophyte is important for them.
Because flowers flowers is where a lot of the phosphate goes, right?
Yeah, you definitely need phosphate for flowers. You need phosphate for your leaves. You need the seed.
Everything.
Everything.
Here's a question. Do we have plant-derived cell wall around the AM branching? We do, right? That's like
it's a cell wall material, but it's not cross-linked, so it doesn't become like a typical cell wall. Work from Paolo Bonfanti's group many years ago showed that it has a structure. They call it a matrix, and they say it has a structure and content similar to a primary plant cell wall, but not one that's become cross-linked. So your normal plant cell walls will be cross-linked and have many different molecules that cross-link and make a tough wall, and that's not there.
So is there still a membrane and then this kind of primary wall?
Membrane, and then there's this what we call a kind of matrix. So a primary cell wall type material is there, and then you have the fungal wall, and then you have the fungal membrane. You've got a sandwich. You've got a membrane in the fungus and a membrane in the plant, and then between the two you've got the wall of the fungus, very thin chitin wall, and you've got this wall-like material of the plant, and they're a wall.
And in between those two things is where they trade.
Well, those two things are really together. The wall of the fungus is kind of within that matrix material.
What is the composition of the membrane that surrounds the hyphae? Is it the same as the plant primary membrane? I think
it's probably the same. We don't know this. It's very difficult to separate. It's difficult to get hold of biochemically. It's not the same in its protein content. So I don't know if you noticed on that picture with the hand in the glove. The glove didn't extend around the edge of the cell, and that's because that protein, the phosphate transporter, is only in the part of the membrane that's around the fungus and not in the part around the edge of the cell. But the lipid content, we don't know too much about. The actual lipid content of the membrane, whether it's the same all the way around.
Yeah, I seem to remember one of our conversations a while ago. Is it, I could be misremembering, but do the lipids go more to like the branches of the fungus, and it's the sugars go more to like the trunk?
Not necessarily, but certainly the transporters that we believe are capturing the lipid, and also the ones that capture the phosphate, tend to be around the finer branches of the hyphae, of the arbuscule. The fungus can probably take sugar even in the hyphae that are running between the cells, because we see that those hyphae, there's evidence they have sugar transporters in them, and there's free sugars in these spaces between plant cell walls. So whilst the fungus is growing into the root, it probably can get a little bit of sugar from this apoplastic space.
That's so neat. How does signaling between AMF and plant root change in phosphorus-rich environment? What drives a symbiosis then? That's a good question. Is there even a symbiosis in a high phosphate environment? Yeah,
so if the plant's growing in high phosphate condition, it won't make and secrete strigalactone. So those first signals will be at a very low level. So the first step is the plant won't be even encouraging the fungus to start to grow in its vicinity. That's one of the control points, but there are other control points in the plant as well. If the plant has already established an association and then receives a bucket load of phosphate, what you see is that somehow internal colonization is restricted. The fungus is not able to spread as much. I don't think we fully know the basis of that, but there's certainly a control there that limits the spread. It may also still involve strigalactones. There are certainly other signaling that goes to the shoots and comes back down, so that the shoot phosphate content is monitored and the amount of colonization in the roots is regulated based on the phosphate content of the shoot, and that has some strigalactone basis in the signaling. But exactly how it's limited, it's not so clear.
Right. Here's one from Eric Richards. Have all AM fungi lost the same FA metabolism gene or genes? Or are there multiple genes and different ones lost in different species?
At least as far as we know at the moment, it's the fatty acid synthase, the FAS complex, that's lost. And all of them have lost that. And they seem to have the same sets of fatty acid biosynthetic enzymes for modifying lipid or making sterols or other decorating lipids, desaturating lipids, cyclizing lipids, anything longer than a C16, a carbon 16 chain length fatty acid, but they can't make that first linear C16 molecule, and that's what the FAS does. The FAS just extends that linear chain 2 carbons at a time and to make a long straight molecule, and those are the ones that the fungi seem to have lost, and they all seem to have lost that. But as I said earlier, there's not so many fungal genomes sequenced so far, maybe a handful, and so maybe there'll be differences when we know more, when we have more genome sequences.
Okay. Yeah, 16 seems kind of big to me. I don't know. Is that, I mean, for a building block? So they can't even make anything below 16.
That's what the genome sequence suggests, and what the predicted enzymes suggest.
Wow,
yeah.
That's amazing. Is it known if nematodes play a role in the fungal plant relationship?
Not this one actually. There are some papers that report a reduction in nematode problems in mycorrhizal plants, that mycorrhizal associations can somehow be protective. And it's unclear whether that's by exclusion because the root is already filled with fungus, or whether there are other mechanisms for suppressing attack by nematodes. But they're not part of it, definitely.
That'd be interesting if that was like another benefit from
It
having
would. It would.
Yeah. Is there a negative interaction if herbicides are applied to tree fruits? I guess to herbicides, would they?
I mean, if you defoliate the plant, herbicide for if you take the leaves off the plant, then I would expect a negative effect because you won't have photosynthesis going on and you won't have extra carbon to go to the roots. But maybe it wasn't herbicide in that sense, or maybe the question was asking whether the herbicide has a direct effect on the fungus in the soil. I'm not quite sure that I followed.
Could be if the herbicide on the above ground, does it impact the relationship below ground?
Only if it would impact the health of the plant. If your herbicide would impact the health of your plant host, then the fungus will suffer too.
That makes sense. If root architecture is changed by AM fungi, does that reduce soil penetration and have implications for reaching soil water? Question.
I think it doesn't stop them growing long, deep roots. It just increases some of the lateral root growth. So I don't think that AM fungi will prevent plants from getting water. And actually, there are some pretty nice studies showing that in some cases, some genotypes again recover from intermittent drought more readily when they have mycorrhizal associations in their roots. So there can be beneficial effects of the fungus on intermittent drought.
Okay, we're getting close to 1 o'clock. Maybe just a couple more questions. Given that fungi's chitin wall is in direct contact with the plant's membrane, is there some kind of specific regulation in order to avoid the activation of defense systems induced by PAMPs?
Yeah, this is a great question. One that we still, the field, when I say we, it's usually I'm meaning the whole mycorrhizal field, still doesn't really understand how the fungus doesn't trigger those kind of PAMP triggered immunity. And some of those PAMPs, some of those chitin molecules are actually needed for the first signaling with the fungus. It's shown that the SERC mutant, if it's somebody who's asking who knows what SERC is, the chitin receptor mutants are not as well colonized as wild type, which suggests that there's actually a positive effect of those receptors and there's a positive effect of being able to detect those short chito-oligosaccharides. So it's a complicated thing. We don't understand why they're not triggering a big PAMP immunity, but it's clear that some triggering is also contributing to the positive signaling for the symbiosis. So work, if you want to read it, it's from Giles Oldroyd's group. Interesting papers.
Would treating trees for fungal disease with fungicide, would that affect the AM fungi?
Fungicides can affect, if they penetrate into the soil and they get to the fungus, it's very likely that they will kill the fungus if they're general fungicides. But you have to consider how much will really get down into the ground and into the soil. I suppose it depends how they're applied.
Yeah, unfortunately, I think we've reached our time. We have many more awesome questions. I apologize for not being able to answer them all for you all. But I want to thank you again, Dr. Maria Harrison, for taking the time today to chat with us. That was absolutely fantastic. That was very enlightening.
Thank you. It was a pleasure to be here and a pleasure to talk with you.
Yeah, and thank you everyone out there who joined us and asked some awesome questions. And I would like to just announce and hope that you could please join us for BTI's next Breaking Ground discussion series, which will feature an interactive chat with BTI's George Jander on Thursday, January 28th, when we'll be talking about cardiac glycosides, which are a class of naturally occurring toxic compounds that have evolved numerous times in many different species of plants and animals, and the animals that have evolved resistance to them. It's going to be a really good one. George has tons of really cool stories around this topic. Some things that I never heard of and never even dreamed were were a thing. So it's going to be a really good one. You can go to btiscience.org for more information and to register for that. And you can also read more about BTI's current research and many other neat stories about BTI science in our 2019 annual report, which you can find online at www.btiscience.org/annual report. And thanks again for joining us and for supporting us. We are an independent nonprofit research institute, and we operate in part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give, or you can email our awesome development team at development@btiscience.org. And thank you so much again for your interest in BTI and your support of the Boyce Thompson Institute. And have a wonderful day and be well. Bye, everyone. Thanks again, Maria.
Thank you, AJ.